Good morning to all, dear ladies and gentlemen, esteemed guests, and valued participants. I extend a warm welcome to each one of you for today's enlightening webinar. I am Dr. Sanjay Nagdev, the co-convener of this webinar, and it is a pleasure to see such a diverse and engaged audience gathered virtually. Today marks the 32nd SPSR webinar, and we are here to delve into intriguing topic of from lab to life, unraveling chemotherapy-induced neuropathic pain with cutting-edge animal models. This event stands as the 32nd in the series hosted by the Society of Pharmaceutical Sciences and Research under the mentorship of Dr. Shashi Alok Sir, the National President, SPSR, and Editor-in-Chief of IJPSR Journal. Our collective aim, inspired by the leadership of Dr. Shashi Alok Sir, is to ensure healthy lives and promote well-being of all at all ages. The objectives guiding our efforts encompass a wide spectrum from holding conferences to promote the pharmacy profession to contributing to programs and services focusing on the health need of India, the prevention of diseases. We also seek to honor eminent pharmacists dedicated to advancing the pharmacy profession and actively promoting the acquisition, dissemination, and the exchange of pharmacy knowledge. It is an honor to have Dr. Vinod Tiwari, sir, Associate Professor in the Department of Pharmaceutical Engineering and Technology at IIT BHU, Varanasi, as our distinguished speaker for today's webinar. Dr. Tiwari's expertise in the field is invaluable, and we eagerly anticipate gaining insights from his wealth of knowledge. In our midst, we are joined by the respected Mrs. Monica Sabarwal, ma'am, convener of this webinar, National Secretary of SPSR and Managing Editor, International Journal of Pharmaceutical Sciences and Research and International Journal of Pharmacognosy. Without further delay, I would like to invite Mrs. Monica Sabarwal, ma'am, to welcome our esteemed guest speaker, Dr. Vinod Tiwari, sir, and lead us through this enlightening session. Over to you, ma'am.
Thank you so much, Dr. Sanjay Nagdev. A very good morning to everyone. I am delighted to welcome all to the 32nd SPSR webinar on From Lab to the Life, the Unraveling Chemotherapy-Induced Neuropathic Pain with the Cut-Edging Animal Models, organized by the Society of Pharmaceutical Sciences and Research, SPSR. This webinar promises to be a groundbreaking exploration into the area of utmost importance in the field of pharmaceutical sciences. I am Monica Sabarwal, the convener of this esteemed event, and it is a truly honor to stand before you today. Our journey into the understanding chemotherapy-induced neuropathic pain will be guided by none other than our distinguished speaker, Dr. Vinod Tiwari, an associate professor in the Department of Pharmaceutical Engineering and Technology at prestigious institute, the Indian Institute of Technology, BHU, Varanasi, Uttar Pradesh, India. Dr. Tiwari, a wealth of knowledge and expertise to this platform, and we are privileged to have him with us today. He will share his insight with us. His dedication to the advancing pharmaceutical sciences and his contribution to research make him an ideal guide as we navigate to the complexities of translating laboratory findings into the real life solutions. The overwhelming response to this webinar is truly heartening. With more than 3,300 registrations pouring in from over 15 countries, we witness a global eagerness to delve into the intricacies of chemotherapy-induced neuropathic pain. This international participation speaks volume about the collaborative spirit within our community and the shared commitment to the advancing scientific understanding. As we embark into this journey together, let us keep in mind the impact of the collective efforts that we can have on the elevating the suffering of those affected by the neuropathic pain. The intersection of research, technology, and compassionate care is powerful space. And through the forums like these, we aim to bridge a gap between the laboratories and the real life applications. I am truly delighted to have our distinguished owner, Dr. Shashi Alok, who is the National President of the SPSR, with us today. His outstanding leadership and dedication to the scientific research have been instrumental in their propelling progress and advancing the culture of innovation within our esteemed community. Under the Dr. Shashiak's visionary guidance, the Society of Pharmaceutical Sciences (SPSR) has witnessed transforming growth and become an excellence in the realm of pharmaceutical sciences. I would also like to express my sincere appreciation for the invaluable contribution of Dr. Sanjay Nagdev, who is the co-convener of this webinar. Their tireless efforts have been a crucial role in transforming a vision of this event into a reality. Behind the scenes, there is a dedication team whose hard work and commitment have been the driving force behind the seamless organization of this webinar. Their attention to detail and passion for the excellence have made this event possible. And I extend my heartfelt gratitude to each and everyone SPSR team. Finally, to all our participants, thank you for being an integral part of this gathering. Your presence enriches the collaborative spirit of this webinar, and we look forward for the vibrant discussions, insights that we unfold today through this event. Without further ado, I invite you to engage, learn, and contribute to the rich tapestry of knowledge that we will unfold over this webinar. Thank you for the eager participation, and let us make the 30-second SP webinar a memorable and impactful event. Thank you so much. Let us begin the 30-second SPSR webinar. Over to you, Dr. Sanjay Nagdev.
Thank you, ma'am, for allowing me to conduct this webinar. Now, let's begin with the brief introduction to Dr. Vinod Tiwari, sir, our esteemed guest. Dr. Vinod Tiwari, sir, is currently working as Associate Professor of Pharmacology in Department of Pharmaceutical Engineering and Technology, Banaras Hindu University. Sir has completed his PhD in pharmacology from the esteemed institute UIPS, Punjab University, Chandigarh in 2012. Before pursuing his doctoral studies, Dr. Tiwari was associated as research associate in Nicholas Piramal Research Center, Mumbai from 2007 to 8, where he contributed immensely in the drug development projects related to pain, inflammation, and arthritis. Dr. Vinod Tiwari carried out his postdoctoral research on neurobiology of chronic pain and its therapeutic targeting at the Johns Hopkins University School of Medicine, USA, from 2012 to 2016. And before joining IIT BHU, Saurav also worked as Assistant Professor of Pharmacology and Toxicology at NIPER, Ahmedabad from August 2016 to March 2018. Dr. Vinod Tiwari is working in the field of pharmacology and neuroscience from past more than 18 years, and his research interest is focused on behavioral and molecular neuroscience dedicated to understanding the various neurodegenerative disorders and the signaling pathways involved so that the better and safe drug candidates can be developed by targeting the disrupted neuromachinery. Dr. Tiwari contributed immensely in the field of neuroscience and pain with more than 80 international publications in peer-reviewed journals, including Nature Neuroscience, the Journal of Clinical Investigation, etc. His research work is rapidly being cited all over the world with more than 4,500 citations and the H index of 38. Currently, he is working on different collaborative projects with investigators from Johns Hopkins University, Russia Academy of Neuroscience, with clinicians and pharmaceutical industries in India. His lab is well funded with grants and funding from SERB, ICMR, and MHRD. Dr. Tiwari has delivered several invited research talks and presenting his research finding in more than 70 national and international scientific meetings. He is also serving as editorial board member and reviewer of many reputed journals. And he is also a grant reviewer for various funding agencies from India, Europe, like Department of Biotechnology, SERB, ICMR, and SethSign Foundation. It brings us immense joy and pride to share that we have garnered more than 3,300 registered attendees from more than 15 countries like Nigeria, Saudi Arabia, India, UAE, Pakistan, Kuwait, etc. And the overwhelming response from such a wide reaching and varied audience is truly humbling. We extend our heartfelt gratitude to each and every participant for choosing to be a part of this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed guest, Dr. Vinod Tiwari, sir, with us. Welcome, sir. I kindly request you to share your PowerPoint presentation and start with your talk. Thank you, sir. Over to you, sir.
Thank you so much, Dr. Sanjay, co-convener of the event. So, a very good morning to all the participants who have joined us from India and other Asia Pacific region, and a very good evening to all the participants who have joined us from other parts of the globe other than Asia Pacific. So, first of all, my heartfelt thanks to the National President of Society for Pharmaceutical Sciences and Research, Dr. Sashi Alok, for inviting me to deliver the lecture in this event. I also convey my thanks to Dr. Monica Sabarwal, who is convener of this event. So I'm really glad to see that the outreach of SPSR is really amazing. Like we have participants more than 3,300 from almost 15 different countries. So yeah, so I will in today's talk I will talk about a new animal model which we have recently developed in our lab at IIT BHU Varanasi. In case if somebody has questions, so feel free to put it in the chat box, or feel free to send your questions to the uh, organizing team of this event, and then once I receive your questions, I will try to get back to you. So I hope my screen is visible to everyone.
Yes, sir, it's visible, sir.
Okay,
You can
perfect.
start.
Very good. So yeah, from lab to life. So that is the title of my today's presentation, where I have tried to address this one of the most common and widespread complication of chemotherapy-induced peripheral neuropathy. So before I talk about this area, I would like to convey my gratitude to Mahamana Pandit Madan Mohan Malviya ji. People who belongs to India, they might be knowing about this personality. He was a freedom fighter of India, and was also the founder of this beautiful temple of learning where I'm sitting today, Banaras Hindu University. So I'm glad to inform you all that recently BHU and IIT BHU has completed its 100 years of existence, and we have recently celebrated the centenary celebration on completion of 100 years. And I'm also happy to share with you that this is one of the largest residential campus of whole Asia. So before we talk about the chemotherapy-induced peripheral neuropathy, the first question, which is very, very obvious to each and everyone who has joined us today: what is pain? Whether pain is good or bad. So you might have heard about IASP. IASP full form is International Association for the Study of Pain. So according to IASP, pain is any unpleasant sensory and emotional experience which is associated with actual or potential tissue damage. Now the very obvious question is that whether pain is good or bad. Most of you will say it is bad. But if I tell you that pain is not bad, it is good. It is protective in nature. So just imagine a situation where you are taking a hot shower of water, and you can't feel how hot is the water. What will happen to you? You will end up burning your entire skin, and may also end up in hospitalization with severe burns all over your body. So it is very, very important to feel and perceive the pain sensation. So pain is ultimately protective in nature. It acts as a warning signal, which sends signals from peripheral nervous system to the dorsal horns of the spinal cord and then to the brain to tell you that something wrong is there and you should take some proactive step to counterattack that damaging stimuli. So now the next question is, how do we feel pain? So you can see on this right hand panel, there's a diagram where a person encounters an injury on his hand. So our peripheral tissues, they consist of nociceptors. We also call them pain receptors. So nature has given us a wonderful gift, just like we do have the taste receptors to taste the different kind of food in our mouth and tongue. We can tell whether a particular kind of food is bitter, salty, sweet, or sour. The similar manner, we can smell different kind of smells, whether it is a good smell or a bad smell. In the similar manner, nature has given us different kind of receptors on our peripheral nerve terminals, which can sense the different kind of pain stimuli. We call them pain receptors. In other words, we also call them nociceptors. So if you see on this left hand side, there are different kind of nociceptors. One of them is thermoreceptive nociceptor, which is responsible for detecting the thermal pain sensation. Like if you're taking a hot shower of water, or if you're holding a hot cup of tea or coffee, this receptor get activated. And the example of these receptors are TRPV1, transient receptor potential vanilloid 1 subtype receptor. And there is another receptor which is temperature sensing. It is known as TRPM8 receptor. So many times you might have eat the menthol. What kind of sensation do we have? We generally feel cold in and around our mouth and tongue. So how this cold sensation we are feeling is because of the TRPM8 receptors activation, which is present in your mouth and tongue. In the similar manner, there are other kind of nociceptors like mechanoreceptive nociceptors, which helps in the detection of mechanical stimuli. If there is some stimuli which is mechanical in nature, for example, some sharp object or any other object which is touching you, then this receptor get activated and sends the signal regarding the mechanical nociception. There is another category of receptor which is also known as chemoreceptive nociceptor. So these receptors are generally activated in response to certain chemicals such as capsaicin. Capsaicin is very common chemical which most of the people, especially in India, use in the spices in the food. So capsaicin is responsible for activation of TRPV1 nociceptor. So if you go to the just 2-3 years back, the Nobel Prize in Physiology and Medicine is given to Professor David Julius for the discovery of this TRPV1 nociceptor, because the discovery of TRPV1 nociceptor has led to the advancements in the understanding of different complex pain pathways and also lead to the development of several therapeutics formulations which are currently in the clinic for the treatment of several painful conditions. Now there is fourth category of nociceptor, which we also call as polymodal nociceptors. As the name indicates, polymodal means these are the nociceptors which can detect more than one painful stimuli at a time. So the example is TRPV1 or TRPA1. For example, TRPA1 nociceptor can detect both thermal and chemical stimuli. So now what happens when a patient, encounters any kind of injury in the peripheral nervous system? It helps in the detection of these stimuli in the peripheral nerve system, and these nociceptors get activated. And the job of these nociceptors is to transmit these pain signals from the peripheral nervous system to the DRG neurons, dorsal root ganglionic neurons, through the different pain fibers like A delta fiber, A beta fiber, and C fiber. If you have gone through the gate control theory of pain, which was given by two amazing scientists, Melzack and Wall, in the year 1965, their work was published in the journal Science at that time, and both these scientists have been awarded with the Nobel Prize because of this amazing discovery of gate control theory of pain. So according to this theory, any non-painful input closes the gate to the painful input. So The fibers which are responsible for carrying the pain sensation from the peripheral nerve terminal to the DRG neurons are A beta fiber, A delta fiber, and C fibers. And this pain information from the DRG neurons, which are the primary sensory neurons, is further transmitted to the dorsal horns of the spinal cord. So in the spinal cord, you know, uh, there are certain modulations which occur, and then the final information, pain information, gets further transmitted through the ascending pathway to the higher pain centers which are present in the brain, like cortex, amygdala, and all of the major brain regions which process this pain information. And brain actually calculates whether this pain or whether this threat is credible enough to take some proactive step or not. For example, if you're holding a cup of coffee, if it is too hot, your brain will tell you to immediately leave it or put it aside. But if the cup of tea or coffee is not that hot, you will decide to hold it and then drink it, right? So accordingly, the brain will tell the body to take the proactive step through the descending pain pathway. So you just imagine all these things are happening within fractions of milliseconds. You just imagine how wonderful is your system. Everything is happening within fraction of seconds and milliseconds. Now the patients who are suffering from this uh, neuropathic pain or any other kind of pain disorders, they mostly complain of two main symptoms. One is mechanical allodynia, second is thermal hyperalgesia. So the question is, what is the difference between allodynia and hyperalgesia? Allodynia is a situation where non-noxious, or you can say a non-painful stimuli become become painful in nature, become noxious in nature. For example, in case of patients who are suffering from chronic diabetic neuropathy, these patients, they generally have difficulty and they feel a lot of pain even when they wear their shirts or when they wear their socks. Do you think wearing the shirt or wearing the socks is a painful condition or a situation? No, not at all. But these patients, they do feel intense pain while wearing the shirt and the pain. So which is a non-noxious stimuli. So this kind of situation is known as allodynia. Now the second condition is that where a certain stimuli is there which is less noxious or less painful in nature, but it becomes more painful in nature in cases where the patients are suffering from certain pain disorders. So this kind of situation is known as hyperalgesia. Now again, if the stimuli is mechanical in nature, we call it mechanical allodynia or hyperalgesia. If the stimuli is uh, some temperature sensing things, then we call it thermal hyperalgesia. Now in this slide, I'm trying to convince you that neuropathic pain is basically a disease. It is not a symptom. Most of us, when encounter, pain symptoms, we just go for the symptomatic relief. But actually, if you try to understand the pathophysiology or the, real cause of that pain, the symptoms which you're observing, then only you can treat those conditions. So there are certain conditions. If you just look at this slide, exposure to certain drugs, for example, anti-cancer drugs or the drugs which are toxic to the nervous system. Alcohol, we all know that alcohol is hepatotoxic in nature, but how many of you know that alcohol also lead to the nerve damage, peripheral neuropathy? This was the area, on which I have worked during my PhD. Now, exposure to certain toxins that also lead to the nerve damage and neuropathic pain. Now, traumatic nerve injury, compression due to certain accidents, viral infection, for example, human immunodeficiency virus, that also leads to the nerve damage and neuropathic pain. Vascular related neurodegenerative disorders, for example, in case of patients who are suffering from stroke associated neuropathy. Nutritional deficiency, we know that our neurons now they are well protected by the myelin sheet, but you know this myelin sheet is a protective covering which is made up of vitamin B1. So in case of nutritional deficiency, especially when vitamin B1 is deficient in your diet, your myelin sheet may encounter the attack by the free radicals which are generating each and every second in your body, in each and every cell of your body, and then they lead to the degeneration of the myelin sheet and lead to the neuronal damage, which ultimately leads to the pain sensation. Certain diseases such as cancer, the patients who are suffering from different kind of cancer, especially bone cancer, they again encounter the severe neuropathic pain symptoms. So cancer act as a double-edged sword where cancer itself is leading to the neuropathic pain, and on the other hand, the drugs which are currently in the clinic for the treatment of cancer, chemotherapeutic drugs, we know that these are one of the highly toxic drugs, but patient doesn't have a choice because doctors want to prolong their life by giving these drugs. But unfortunately, these drugs they lead to a lot of side effects and toxicities in these patients. And peripheral neuropathy and neuropathic pain is one of those symptoms. So that is, the work which I'm going to talk with all of you today. Metabolic disturbances, for example, in case of patients who are suffering from diabetes, hyperglycemia, that again leads to the free radical generation, synthesis of advanced glycogen end products, which ultimately leads to the nerve damage and neuropathic pain. Surgical procedures, amputations, in case of soldiers, military veterans, or certain cardiac conditions where doctor has to perform the surgical procedures and amputations in certain patients. During those procedures, it leads to the nerve damage in these patients, which again leads to the neuropathic pain for rest of their life. So neuropathic pain is not a symptom, it's a disease which needs the proper treatment. Now, in this study, we have tried to develop a new model of chemotherapy-induced peripheral neuropathy, and I will go to that slide like why we have thought about it. But before that, why animal models are important in the field of drug discovery. We all know that, animal models are the valuable tools for testing the potential therapies and the drugs. We cannot test any new molecule straightforward on the humans because of their safety concern. We all know from the history that there are certain instances where certain drugs were not properly tested in the animal models or other in vitro system, and they came to the market without proper testing, and the result we all know. One of the example is the thalidomide disaster, where one of the drug which was consumed by pregnant women throughout the Europe to control their morning sickness. That drug was not properly tested in the preclinical models, and ultimately, the result is that thousands of children all over the Europe were born with a disease, genetic abnormality, which we all call as phocomelia, where the children were born with seal-like limbs, without proper development of their body parts, and those children and their families are still suffering after so many years. Even later on, the company came and made the apology and given some compensation always, but they can never compensate for their suffering through which they are going. So yeah, so proper drug testing in animal model is very, very important before any drug progresses to the clinic. Now, there are several genetic manipulation which we can easily do in the animal model, and we can mimic the human situation. This can allow the researcher to study the effect of genetic variations on the disease. Time efficiency, animal models can provide us quicker result, allowing the scientists to test the hypothesis and develop the treatments more rapidly than in the human trials. We all know that clinical trials is a long-term process. It sometimes goes from 10 years to even to more than 12-15 years. Safety concerns we have already discussed. Translational research, we all know that once any molecule is found safe and efficacious in the animal model, then only we can go for the clinical trial to test their further safety and efficacy in the human volunteers, healthy volunteers first, and then in the patient subset. First in the small number of population, and then in the larger cohort of patients using the multicenter trials in the phase 3 clinical trials. Now, the situation or the problem is that, if you are picking the wrong preclinical model, most of the drugs, are bound to fail in the clinical trial. And we all know that the drugs, which generally passes through the preclinical drug testing, out of those drugs, more than 90% of the drugs, they fail in the clinical trials. And one of the major reasons for that is testing of those molecules in the wrong preclinical models. So for several decades, preclinical scientists have been screening the novel molecules with the potential to treat several neurological disorders, but unfortunately the translation of this promising compound from preclinical to clinical phase has been very, very low. And there are several complex reasons for this, but one major factor is the choice of the wrong model, wrong animal model. If I specifically talk about the chemotherapy-induced neuropathic pain model, we all know that right now the scientists are using monotherapy-induced neuropathy pain model, where they are giving one of the anti-cancer molecules such as paclitaxel, cisplatin, or any other anti-cancer drugs, and they're inducing this neuropathic pain symptom in these animals, and then they are giving their test molecule to see whether it can suppress this anti-cancer drugs induced neuropathic pain or not. So most of the preclinical models are monotherapy based right now in the preclinical phase, what scientists are using. But when I was talking to one of the clinician here in Institute of Medical Science, BHU, I came to know, the in patients mostly, who are, being treated for the different kind of cancer, they are not being treated with one anti-cancer molecule, but they are being given the treatment with more than 2 or 3 anti-cancer drugs at a time for the so that we can treat their cancer. So that toxicity which they are observing, and peripheral neuropathy is one of them, is a result of not just one anti-cancer drug, but it is a result of more than 2 or 3 anti-cancer drugs. So we thought of developing a new animal model of chemotherapy-induced neuropathic pain, where we can utilize the combination of at least 3 chemotherapeutic drugs, and then we compare the effects with a conventional paclitaxel model using rats. So if we talk about the prevalence of the chemotherapy-induced neuropathic pain, more than 10 million deaths in 2022 is reported because of cancer, and it accounts to almost 15 million in, if the trend continues, it will lead to 15 million in 2040. And we all know that chemotherapy is one of the most widely used therapeutic approach for that. But unfortunately, it leads to a lot of side effects, toxicity, and neuropathy is one of them. Unfortunately, the patients who are on chemotherapy, almost 80% of those patients, they develop the chemotherapy-induced neuropathic pain. So you can just, imagine the prevalence, the huge prevalence of this side effect and toxicity associated with the anti-cancer drug. So you can see this, pie chart as well, which suggests that, like the patients who are taking cisplatin, it leads to the 25% of the chemotherapy-induced neuropathic pain cases are because of cisplatin. 38% cases of peripheral neuropathy is because of paclitaxel administration, and others are also contributing to the chemotherapy-induced peripheral neuropathy. Now, what is the mechanism of chemotherapy-induced neuropathic pain? So it's a double-edged sword. Why? Because whenever a patient is taking the anti-cancer drugs, there could be certain nerve damages because of the proliferation of the cancer cell inside the patient body. And at the same time, when these patients are being treated with the different anti-cancer drugs, that also lead to the activation of different, uh, you know, pathways in which the ion channels activities are altered, such as, drugs like oxaliplatin, vincristine, they lead to the activation of the glutamate receptor, which further leads to the influx of sodium ions, calcium ions, and then at the same time, it also lead to the uh, oxidative stress generation of free radicals, which leads to the mitochondrial damage, and both these cascade they further leads to the increase in pro-inflammatory cytokines like TNF alpha, interleukin 1 beta, interleukin 6, nitric oxide, and they all collectively lead to the activation of caspases and ultimately lead to the neuronal cell death. And all this contribute to the neuronal hyperexcitability and peripheral sensitization, which leads to the continuous awareness of the neuropathic pain in these patients for many, many years, not for few days or months, but for many, many years because of the damage to the A beta fibers, C fibers, and then peripheral sensitization of the nociceptors. And these patients, they generally complain of numbness in their extremities like hands and the feet. Sometimes they complain of the tingling sensation, like somebody is, pricking them with the needles, this kind of pain sensation. Sometimes they also complain of the cold sensitivity, which is one of the most common symptom of the chemotherapy-induced neuropathic pain. And unfortunately, if we talk about the drug treatments, so far we do not have any effective treatment or US FDA drug for the treatment of chemotherapy-induced neuropathic pain. Okay. So in this study, as I mentioned, we thought of developing a new model, but there are certain conditions. When you are developing a new model, you need to validate the new model by using different kind of validities, including predictive validity, face validity, and the constructive validity. So in the face validity, you have to see whether the animal model which you have developed is replicating the same kind of behavior symptoms, just like it is replicating in the human beings. At the same time, you also need to see that whether these symptoms, they are responsive to the standard pharmacological therapies which are currently available in the clinic. So that is known as the predictive validity. And after completing these two validities, finally, one has to check their model for the constructive validity, where one has to check whether the model they have developed, is replicating in terms of the biomarkers which are involved in the in any kind of disease. In this case, the chemotherapy induced peripheral neuropathy, whether those markers are down regulating or up regulating. So that is also known as the mechanistic validity. So once your model is uh, responding to all these three validity tests, you can say that, okay, this is the model which can be used for the screening of new drugs. So in this study, we have used the male sprout dolly rats weighing from 200 to 220 gram. And we have used paclitaxel to induce the monotherapy induced chemotherapy pain model. And a combination chemotherapy model, we have used the combination of three anti-cancer drugs. First, paclitaxel at the 2 mg per kg, cisplatin 2 mg per kg, and vincristine 0.5 mg per kg by intraperitoneal route. And all these drugs have been picked based on the literature review, clinical and preclinical. And gabapentin was the another drug which has been used as a standard drug in this model, just to uh, confirm the face validity model. And then we have performed the different behavior tests for the assessment of pain in these animals. So Hargreaves test has been used for the assessment of thermal pain behavior. Warnfree test has been used for the measurement of mechanical allodynia. Pinprick test has been used for the measurement of mechanical hyperalgesia. And acetone spray and cold plate test were used for the measurement of cold allodynia and cold hyperalgesia in these animals before and after the induction of chemotherapy-induced peripheral neuropathy. And then we have observed these animals for their daily body weight changes and also for the mortality in these animals. And after the completion of the study, we have sacrificed the animals and we have collected their sciatic nerve for the measurement of different biochemical parameters, including increase in their malonaldehyde levels, reduced glutathione and catalase level, which are antioxidant enzymes, which are protective in nature. And then we have also harvested their dorsal root ganglia, DRG neurons, which are primary sensory neurons and plays a very, very important role in the pain transmission from peripheral nervous system to the central nervous system to the spinal cord. So, and then we have also harvested the spinal cord, which acts as, which consists of secondary sensory neuron and plays a very, very prominent role in the transmission of pain information or pain signal from spinal cord to the brain in animals. And then we have performed the real-time PCR analysis for the measurement of mRNA expression of different biomarkers which are involved in chemotherapy-induced neuropathy, and then the protein expression of different biomarkers. So let's talk about the first set of data where we have measured the body weight of the animals. If you look at this data, there are three different groups. First is the vehicle group, in which we have given the vehicle of the drugs in which these two drugs have been prepared, have been dissolved, which is represented with the blue circle. In the second set of group, we have given the paclitaxel alone administration, which is represented with the red circle. And then the third group is the combination chemotherapy, which represent the purple circle. In this group, we have given the combination of three different anti-cancer drugs. And then we have monitored the body weight of these animals before the induction of chemotherapy, which is BL baseline, and then at different time points after giving the chemotherapy, that is up to day 56, that is almost up to 8 weeks after giving the chemotherapy. And it is very, very clear from this set of data that the animals which were treated with vehicle and paclitaxel alone did not had any effect on their body weight. So their body weight is almost, consistent, is not changing. But the animals which were treated with combination chemotherapy, means three different anti-cancer drugs, their body weight has been drastically decreased. And it was a significant decrease from starting from 2 weeks after the combined chemotherapy administration, and it persisted for almost 8 weeks. So which is one of the most, I would say, the prominent sign of the chemotherapy toxicity in humans as well. The patients who are on anti-cancer, they gradually lose their body weight. So that was the first sign which told us that, okay, this model, might be more closer to the human situation. Then we have measured the mechanical allodynia in these animals. On the right hand panel, you can see this. The animals are placed on the one-way wire mesh, which is a stainless steel wire mesh, and they have been poked with one-way filaments. And there is a set of filaments, ranging from very low force like 0.2 gram, which is almost thinner than your hair, and then it goes up to 15 gram, which is the maximum force which we have used. It's a highest diameter filament which we are using in the rodents. And then we are testing the paw withdrawal threshold of these animals. So we are measuring at what force the animals are withdrawing their paws. But you should be experienced enough to understand, whether this is a painful response or it's just a movement of the animal, locomotion of the animal. So you need a lot of experience to judge between these two different kind of behaviors. So if you look at the baseline data before the induction of chemotherapy, in all the three different groups, vehicle, paclitaxel alone, and the animals which were treated with combination of anti-cancer drugs, the baseline paw withdrawal threshold is almost 20 to 22 gram. But when these animals were treated with paclitaxel and combination chemotherapy, their paw withdrawal threshold has significantly dropped from 22 gram to almost 10 gram, and then finally to 4 to 5 gram on after 1 week of chemotherapy administration. And then if you look at this data, the animals were continuously showing the pain behavior, which persisted for almost, day 28 in case of paclitaxel alone treated animals. But the interesting part which we observed in this study is that the animals which were treated with combination chemotherapy with anti-cancer, combination of anti-cancer drugs, their pain was lasted for prolonged duration of time. You can see, their pain lasts for much more longer duration than the paclitaxel alone model. And they were showing a significant pain even 7 weeks over the chemotherapy administration. So that means there is a increase of almost, if you look at like 3 weeks, that means almost 21 days. They have pain for this much longer duration as compared to monotherapy model. Then we also measure the pain behavior in the right paw. So both left hind paw and the right hind paw of the animals. So it also shows the similar kind of trend in these animals. Then we have measured the thermal pain behavior in these animals because of thermal pain stimuli. So for this, we have used the Hargreaves apparatus. In the Hargreaves apparatus, if you see on the bottom, there is a device which can, give the IR radiation to the animals supplantar region of their hind paws. And then we are measuring at what time the animals are abruptly withdrawing their paws in response to the IR stimuli. So we are using the intensity which is generally not painful in the healthy animals, but if the animals are suffering from some kind of disease, they will immediately respond to this IR radiation. So we are using the constant intensity of the IR in this test. So if you look at this data both in the left hind paw and the right hind paw, the paw withdrawal latency, the time duration was almost around 17 to 18 seconds both in the left and the right hind paw. But after the induction of chemotherapy, almost on day 4 to day 8, there was a significant drop in their paw withdrawal latency. Surprisingly, again, in the Hargreaves test as well, we found that the animals on paclitaxel alone treatment, they recovered much quicker than the animals which were treated with combination chemotherapy. So in the cocktail model, on the combination model, you can see the animals was showing significant increase of their pain behavior to almost 7 to 8 weeks after the chemotherapy administration, both in the left hind paw and in the right hind paw. So this is indicating that there is something going on in these animals. You can say maybe more severe toxicity, or some changes in their molecular expression on nociceptors. Then we all repeated the another behavior test, which is one of the most characteristic symptom of chemotherapy induced pain, which is cold allodynia. The patients who are suffering from chemotherapy pain, they mostly complain about the cold pain. They have a lot of pain even when they open the refrigerator in their house. So to measure their cold allodynic behavior, we have used the acetone spray test, where we are putting the animals again on the one frame mesh, which is made up of stainless steel, and we're spraying the acetone with the help of syringe on their plantar surface of their hind paws, and then we are measuring the response score in these animals. So again, if you look at this data, before the induction of neuropathy, these animals are having a very low response score. But as after like almost 1 to 2 weeks, the pain response score was significantly elevated in these animals. Again, we have observed a right shift in the pain behavior of the animals which were treated with the combination chemotherapy. It's a huge difference if you compare between paclitaxel alone and the combination chemotherapy groups. So those who don't know about the trip channels are the most important ion channels which express in the in the peripheral nervous system, peripheral nerve terminals, and also express in the central nervous system. And these trip channels has the capability of like regulating the flow of different ions, and also has the capability of sensing the different kind of stimuli, as I have discussed in the first part of my presentation. So there are different kind of trip channels like trip V1, trip A1, and trip M8. So trip V1, as I mentioned, I'm repeating again, is responsible for the sensation of noxious heat and cold heat stimuli. Trip A1 is mostly responsible for the detection of mechanical stimuli, and trip M8 is responsible for the detection of cold stimuli, like when you eat the menthol or something, cold, whenever you are touching any ice surface. So there is activation of this trip M8 nociceptor. So after completing the behavior studies, we wanted to understand what is happening with the strip channels in the DRG and in the spinal cord, because DRG, as I mentioned, are the primary sensory neurons which helps in the transmission of pain information from peripheral nervous system to the central nervous system. Then we were also interested to understand what is happening at the with these strip channels at the spinal cord level, the dorsal horns of the lumbar spinal cord. So as you can see in this data, the TRPA1 mRNA expression were uh, increased. There is a trend in case of animals which were treated with paclitaxel alone, but this increase was not significant. NS means non-significant. But when the animals were treated with combination chemotherapy, with a combination of 3 anti-cancer drugs, there was a significant elevation in the TRPA1 mRNA expression, both in the DRG and in the spinal cord of animals. Then we have also measured the mRNA expression of TRPV1. Again, we found that TRPV1 expressions were not significantly elevated. There was certain trend, definitely in case of monotherapy induced pain, which is paclitaxel induced neuropathy, but that trend was not significant enough. But in case of animals who were on combination chemotherapy, they show a robust increase in their TRPV1 mRNA expression. Again, both in the DRG neurons and in the spinal cord, lumbar region of the spinal cord of these animals. Then we went on to measure the expression of another TRP channel, which is TRPM8. We observed a similar kind of trend where, animals on combination chemotherapy showed a drastic increase in the TRPM8 mRNA expression, both in the DRG and in the spinal cord. And all these studies were done on day 35 after the chemotherapy administration. Then we went on to measure the another important marker, which is responsible for the hyperexcitability in the DRG and the spinal cord, NR2B, which is a subunit of the NMDA receptor. So it is already reported that whenever there is, increased neuroinflammation, there is, increased expression of the NR2B, both in the DRG and in the spinal cord, which contributes to the increased hyperexcitability of the neurons, which ultimately leads to the central sensitization and leads to the awareness of the chronic pain in the patients. So we wanted to understand if these TRP channels are contributing to the increase in NR2B or not. Again, we found that the animals which were treated with paclitaxel, they showed no significant difference in the NR2B mRNA expression, both in the DRG and spinal cord, but combination chemotherapy leads to the significant increase in the NR2B mRNA expression in both these tissues. Then we went on measuring the other important biomarkers involved in neuroinflammation and pain, the two neuropeptides, substance P and CGRP. So I will not spend much time here, but we observed a similar kind of trend here as well, where paclitaxel administration does not lead to significant increase in either substance P or CGRP, but administration of combination chemotherapy lead to a substantial increase in the CGRP and substance P in both the DRG and spinal cord of the rats. Then we went on measuring the neuroinflammation in these animals. So TNF alpha, tumor necrosis factor alpha, which is one of the most important marker involved in neuroinflammation, was measured in the DRG and spinal cord of these rats. And surprisingly, we found that in case of paclitaxel treated animals also, there was a significant increase in the TNF alpha, both in the DRG and the spinal cord. And when we treated the animals with combination chemotherapy with 3 different anti-cancer drugs, that increase was further elevated. Then we went on to measure the another neuroinflammatory marker, which is interleukin 1 beta, and we observed that the animals which were treated with paclitaxel alone did not show any substantial increase in the IL-1 beta expression. But the animals which were treated with combination chemotherapy, they showed a significant increase in IL-1 beta expression in both in the DRG and in the spinal cord of the rats on combination chemotherapy. So up to then, we understand, we got an idea. Okay, so if you treat these animals with the combination chemotherapy, you will find a pain behavior which is, prolonging for much longer duration of time, for almost 3 weeks longer than the conventional paclitaxel model. And then we also found that the different markers, including TRP channels, neuroinflammatory markers, and also neuropeptides, they were significantly elevated, specifically in the animals who were on combination chemotherapy. So in the next step, we went on to understand, to prove the another kind of validity, which is known as pharmacological validation, or you can also call it face validity. If we treat this animal with some standard pharmacological treatment, can these symptoms be recovered in these animals? So we have followed the same protocol, where we have acclimatized the animal for first couple of days, for a week, before starting the pain behavior baseline. Then we measured their pain behavior baseline, followed by the treatment with the anti-cancer drugs. After that, we have treated these animals with the gabapentin at a dose of 60 mg per kg. And then we have measured their pain behavior at 2 weeks after chemotherapy administration. So we have chosen that time point because if you look at the behavior data right over here in the previous slide here, the animals, these animals which were treated with paclitaxel alone and combination chemotherapy, they have showed a peak a response, or you can say peak pain score at almost 2 weeks after the chemotherapy administration. So when we started, treating these animals with gabapentin, we picked this time point as a time point for the treatment, because if we are treating the animals or patients in a phase where their pain is resolving by itself, then it doesn't make any sense. Patient generally goes to the doctor when they are suffering from pain the most, right? So we have chosen the 2 week time point as a time point where we have treated the animals with gabapentin, and then at the end of the study we have sacrificed these animals, and then we have collected their tissues like sciatic nerve, DRG, and spinal cord, and then we have measured the different biochemical and molecular markers in different tissues. If you look at this data here, in the Hargreaves test, we have measured their thermal pain behavior, and here you can see 5 different groups. First blue circle is the vehicle treated animals, where we have given the vehicle for the PTX and the combination chemotherapy. So these animals, you can clearly see their baseline is almost stable up to 2 weeks. So there is no change in their pain baseline. But the animals which were treated with paclitaxel, red circle, and combination chemotherapy, which is purple circle, their povidone latency was significantly dropped on day 2, day 4, and especially on day 8 after giving the this chemotherapy administration. While the animals which were treated with gabapentin in both PTX group, this green circle, and then this light green circle, which is combination chemotherapy with gabapentin, you can see their pain behavior was, or you can say their povidone latency was somehow decreased, increased, as compared to the animals which were treated with anti-cancer drugs alone. So this indicates that gabapentin is able to restore their pain behavior a little bit, but there is not a complete recovery. It is still in between, because none of the drugs are miraculous drug. They can treat the pain, but not completely. So that is the biggest, a problem with most of the drugs which are currently in the clinic. They are not very efficacious. They only provide the partial relief, and same is true in case of gabapentin as well. So then we have measured this pain behavior in the right paw as well, and then you can see, again, gabapentin was able to restore the pain behavior in these animals to certain extent. Then we have measured the mechanical pain behavior of these animals by using the Von Frey hair test, and again we found that the animals which were treated with paclitaxel and combination chemotherapy, their povidone threshold was significantly decreased over a period of time. And when these animals were treated with gabapentin at a dose of 60 mg per kg by intraperitoneal route, their povidone threshold was significantly increased from 5 gram to almost 10 to 12 gram, which indicates that gabapentin has some effects in these animals, including the animals which were treated with the combination chemotherapy, both in the left paw and in the right hind paws of these animals. Then we went on to further measure the expression of different, TRP channels and the neuropeptides as we did in the previous study. And then now you might be surprised to see that in this study, the animals which were treated with paclitaxel, they also show a substantial increase in the TRPA1 mRNA expression. Okay. Now you may ask a question, why we observed a TRPA1 mRNA expression, in this study and not in the previous study. But if you look at the time point here, we have sacrificed these animals at day 14. But in the previous study, we were sacrificing the animals on day 35 after the chemotherapy administration. So of course, at 2 weeks when the pain is at peak, even in case of animals which were treated with monotherapy like paclitaxel alone, they also show a substantial increase in the TRPA1 mRNA expression, both in the DRG and the spinal cord. So panel A is the DRG data and panel B is the spinal cord data. But when these animals were treated with gabapentin, the expressions of TRPA1 were significantly decreased. And then we went on measuring the other TRP channel, which is TRPV1, which contributes to the thermal hyperalgesia, thermal pain behavior. We found that in both paclitaxel and combination chemotherapy treated animals, there was significant elevation in the TRPV1 mRNA expression, both in the DRG and in the spinal cord, which was significantly attenuated on treatment with gabapentin 60 mg per kg dose. Then we went on measuring the third TRP channel, which is TRPM8, which is mostly responsible for the cold pain behavior in the patients suffering from chemotherapy pain. We surprisingly found that in case of paclitaxel treated animals, we did not found any increase in the TRPM8 mRNA expression. So TRPM8 expression were selectively increased in case of combination chemotherapy model. So that was I would say, a difference between these two models which we observed, at 2 weeks after chemotherapy administration. And when we treated these animals with gabapentin, there was a sharp decline in the TRPM8 mRNA expression, again both in the DRG and in the spinal cord of these animals. Now we further went on to measure the protein expression of NR2B, which is the subunit of the NMDA receptor, and another important marker, which is a marker for the glial cell activation, IBA1. So if there are increased expression in the IBA1 in any tissue, it suggests the increased macroglia activity. And if there is increased macroglia activity, that may lead to the increase in neuroinflammatory cytokines in that particular tissue. So interestingly, when we subjected both the DRG and the spinal cord of these animals to Western blotting to measure the protein expression, we found a significant upregulation in the NR2B protein expressions and IBA1 protein expression as well, both in the DRG and in the spinal cord of the animal. This elevation was really robust, as measured by the Western blotting. And then when these animals were treated with gabapentin 60 mg per kg, both NR2B protein expressions and IBA1 protein expression were significantly down-regulated, which indicates that gabapentin can decrease the glial cell activity and NR2B central sensitization both in the DRG and the spinal cord. Then we went on measuring another important marker, which is the marker for the cell integrity, cell membrane integrity, and it is ICAM1, which is intracellular adhesion molecule 1. Now if some of you knows about ICAM1 protein, this is a protein which talks about the blood-brain barrier integrity. So increased expression of the ICAM1 increases the blood-brain integrity of the any species like animals or humans is getting compromised. So anything from the peripheral nervous system then can easily penetrate into the blood-brain barrier and may lead to the upregulation in other factors. So here we found that when these animals were treated with paclitaxel and combination chemotherapy, there was a significant upregulation in the ICAM1 protein expression in the DRG and the spinal cord of these animals, and which was significantly down-regulated on treatment with the gabapentin. So this is the overall summary from this study, where we found that the treatment of paclitaxel, leads to the mechanical allodynia, thermal hyperalgesia, and also the combination chemotherapy also lead to these symptoms, but the pain lasts only up to 35 days in case of paclitaxel model. But in case of animals which were treated with combination chemotherapy, the pain lasts for much longer, which is almost day 56, which is almost almost 7 to 8 weeks after the chemotherapy administration. So this talks about the phase validity of the new model. And then we have treated these animals with a standard pharmacological treatment which is available in the clinic, gabapentin at a dose of 60 mg per kg, which was able to attenuate some of these symptoms, not all, and to certain extent only, which talks about the predictive validity of the model. And then finally, we have proven the constructive validity of the model by measuring the different biomarkers associated with glial cell activation, neuroinflammation, and central sensitization. So all these different kind of validity prove that this new model, is important and can be, used for the screening of new drugs. So, so we personally feel that this novel model demonstrates a phase predicting constructive validity and can enable us to further characterize putative drug to treat the CINP, and also can help us to understand the neurobiological mechanism of the same. And I'm very proud to say that this work I have presented last year, not last year, actually, yeah, last year actually, in the International Association for the Study of Pain meeting in Toronto, Canada, where we have received so much appreciation on this model that at least they have encouraged that at least somebody has thought about this kind of a study, because all over the world, people are currently using monotherapy based model for the screening of drugs for treatment of chemotherapy induced pain, but none of them are thinking that this complication is not a result of one anti-cancer drug but is a result of at least 2 to 3 anti-cancer drugs. So our lab is just working in this area where we are trying to develop different kind of model with different kind of combination of these drugs to see if there's any difference, including the gender differences. So this work was recently accepted for publication in one of the journal. Now, based on these findings, we thought that we can we develop certain drugs or therapeutics for the achieving the analgesia without the adverse effects, because most of the drugs which are currently in the clinic, including gabapentin or opioid like drug like morphine, they lead to the lot of side effects and toxicities, including respiratory depression. We know that there is a term which is known as opioid crisis, where patients died because of the overdose of the opioid due to a lot of side effects, including respiratory depression. Sometimes it also lead to the motor incoordination in the patients. So when these patients are on these drugs, doctors strongly suggest them not to drive, not to operate any heavy machinery, because the patient may get sedation or may get dizziness and may end up hurting themselves, which sometimes may cause the life of the patient as well. So these are the certain serious issues, including drug addiction as well in these patients. So we thought, can we develop some uh, new therapies for the treatment of this chemotherapy induced pain in by utilizing this new model which we have developed in our lab. So to answer this question, we have investigated the effect of one of the new molecule, I would say burgeonin, which is not very well studied in the preclinical studies. It's a natural polyphenol, in the newly developed model of chemotherapy neuropathic pain. So burgeonin, for those who don't know about this molecule, it's a 4 or methyl gallic acid, and it is mostly found in the plant, belonging to the species Virginia ciliata and Virginia ligulata. So we thought of using this molecule because there are certain studies which suggest that burgeonin may have a potential protective effect in inflammatory pain, and it may have an effect on MAP kinase and CDK5 expression in the LPS treated rats. So these were the findings which prompted us to look after this molecule, whether this molecule can treat the chemotherapy induced peripheral neuropathy or not. So after this, we have repeated the same model where we have treated the animals with the combination chemotherapy, that is a combination of 3 anti-cancer drugs, and then we have treated these animals with 3 different doses of burgeonin at a dose of 25 mg per kg, 50 mg per kg, and 100 mg per kg. And as I mentioned in my previous slides, we have taken the time point of 2 weeks after the chemotherapy administration, because that is the time point where the pain of the animal is at its peak. So after 2 weeks of chemotherapy administration, this is pre-chemo baseline, where you see none of these 6 groups are not showing any kind of pain behavior. But after the administration of combination chemotherapy, all the 5 groups excluding naive. Naive means healthy groups in which we have not given the anti-cancer drugs. So all the 5 groups, their povital latency has significantly dropped to almost 2 to 3 seconds. And when these animals were treated with different doses of burgeonin, there was a partial restoration of their povidol latency. And this red line, you can see it's a vehicle treated group where vehicle is not producing any beneficial effect to these animals. The same study we repeated in the right paw just to confirm, what is happening with the other paw of the animals. Then we have calculated the percent maximum possible effect of these animals, and then we measured the cold pain behavior in these animals using the ice flow test. In the ice flow test, we keep the animals, on an ice floor which is maintained at 0 degrees Celsius, because as I mentioned previously as well, cold allodynia and hyperalgesia are some of the characteristic symptoms associated with the patient suffering from chemotherapy-induced peripheral neuropathy. So you can see that in these animals, the duration of licking of their paws, because when they are put on an ice floor, the animals who are suffering from this chemotherapy-induced neuropathy, their duration of licking is bound to significantly increase. So you can see the duration of licking has been increased from 1 second to almost 7 to 8 seconds. And when these animals were treated with different doses of burgeonin, there was a dose dependent decrease in their licking duration. Although this restoration is not that impactful, I would say, but still this was able to decrease the duration of licking in these animals. And the similar kind of data we found in case of animals which were treated with on the right hind paw. But you can definitely see gabapentin is much more effective as compared to burgeonin at a dose of 60 mg per kg. Then we have measured the acute pain response of the animals which were treated with burgeonin, because it is very, very important to understand, whether a molecule is acting as an analgesic or as an antinociceptive. Opioid-like drug, which are centrally acting analgesics, they have a strong antinociceptive and as well as analgesic effect. So sometime, it is very important to understand, how your molecule is behaving in the living system. So to measure this kind of response, we take the naive animals which are not suffering from any disease, and these animals were treated with different doses of burgeonin, because you don't want any molecule to alter the normal pain threshold of the patients or the animals. So we found that the animals which were treated with gabapentin and different doses of burgeonin, they did not show any effect on the latency to tail flick. So this we have used by using the tail flick test, where the tail of animal is exposed to the IR radiation at particular intensity. And then we have also calculated their percent maximum possible effect. And then we have also performed the tail clip test, which is also another measure for the measurement of acute pain response in animals, where we are measuring the latency by the animal to attack the clip attached to its tail. So we found that both gabapentin and burgeonin at different doses does not lead to any changes in the latency to tail flick and latency to attack clip, which clearly indicate that this molecule behaves more as a antinociceptive rather than analgesic. Now, if we talk about the field of pain research, there is a lot of debate, which is always going on among the pain researchers all over the world. Whatever we are measuring in animals, whether it is a true reflection of the pain phenotype in humans. So most of the behavior which I discussed with you so far are mostly based on evoked pain behavior. Now, what do you mean by evoked pain behavior? Evoked pain behavior means we are providing certain kind of stimuli to the animals, and then we are measuring the pain response of these animals. For example, in case of one hair test, we are providing or we are poking the paws of the animals with one filaments. In case of Hargreaves test, we are providing the IR radiation. So all of these tests are associated with certain kind of stimuli. But the debate is that, the patients who are suffering from chronic neuropathy or other kind of chronic pain disorders, they do have the pain even when they are at rest, when they are not working, when they are not perceiving any stimuli from outside. So are we measuring the right pain behavior in animals, which is mimicking the clinical situation? So to answer that question, one of the scientists, Professor Frank Poreka, I met him, when I was in US. I attended one meeting, International Narcotics Research Conference. It was held in Phoenix, Arizona in the year 2015. So I met him there, and his lab has done a very good work in the field of spontaneous ongoing pain. So where they have developed this new model, which is known as condition place preference, as an alternative measure for the measurement of spontaneous ongoing pain in animals. So we all know that pain is aversive in nature. So this condition place preference has previously been used in the drug addiction studies, just to measure whether certain substances are addictive in nature or not. So for the drug addiction, let me explain you a bit. They are putting the animals in different chambers, and then they are measuring, in the post conditioning, they are restricting the animals and then treating with the vehicle and with the addictive drug. And then in the post conditioning, the animals are free to go to any chamber. Then they are measuring how much time the animal is spent in particular chamber. So if the animal is spending more time in the chamber where he has been given the addictive drug, that means the substance is having a addiction potential. So this scientist, I would say, was very smart. He used this apparatus, just to measure the spontaneous ongoing pain in a different way. So we all know that pain is aversive in nature. So whatever we are measuring in these animals using condition place preference is because of their preference to certain chamber, which is associated with certain experiences of those animals. Now, if we talk about the pain, we all know that pain is aversive in nature. Nobody likes the pain, right? But if you can treat the pain of these, patients or the animals in particular chamber, that will lead to the feeling of pain relief, which is rewarding in nature, right? And there is an activation of reward circuitry in the brain, such as ventral tegmental area, nucleus accumbens, which means there is a release or surge of dopamine in these brain regions, and that leads animal to feel good in that particular chamber. So animal can clearly differentiate, between the chamber where the animal did not receive the pain relief or did receive the pain relief. The concept is same as that of drug addiction, but the only difference is that here the animals are treated in the morning with the vehicle in one chamber, which is having a different, visual and the tactile cue. If you look at these chambers, one chamber is having a vertical stripe, another chamber is having a triangular stripe or dotted designs. So we are providing different kind of visual cues in both the chambers, in different chambers. And at the same time, the floor of these chambers are different. One is plain, completely plain, and another is having the grooves, so animal can feel while walking on the surfaces of these platforms. So animals are having two different kind of cues here. One is visual cue and another is the tactile cue. So animals can clearly differentiate between both the chambers when they are being treated with the vehicle drug and when they are being treated with the analgesic drug, a strong analgesic. So this relief of pain is rewarding in nature, which is reflected in terms of increased preference to that particular chamber. So if you look at this data, so here we are measuring the time spent in the different chambers by the animals, and all this data is is recorded by the video tracking software, and everything is automated. No interference by the experimenter. It's completely automated. So what do you see here? The red color bar are the time spent in the saline paired chamber, and we are measuring the preference of the animals before the conditioning. Conditioning means when we are restricting these animals in one of these chambers. So in the preconditioning and the post-conditioning, animals are free to move to any chamber, and that activity is being recorded and monitored. So in the preconditioning, when they are not conditioned with any treatment, you can see the animal did not have any preference for either saline paired chamber or the burgeonin 100 mg per kg. Here we have used the highest dose of the burgeonin. So there is no preference between these two groups. But in the post conditioning, if you see, after conditioning the animal with the different treatment, with the vehicle and with the drug, the time spent in the saline paired chamber was significantly decreased, while the time spent in the burgeoning, or you can say the drug treated chamber, was significantly increased, which clearly indicate that and these animals, let me tell you, these animals are suffering from chemotherapy induced neuropathy. That means these animals are having pain. So because this is rewarding after giving burgeoning, that's why the time spent in this chamber is increased. Now you can argue with me and can ask me, okay, if this drug is leading to the increase in time spent in drug paired chamber, it may be a addictive behavior as well. The drug may not be acting as an analgesic, rather it might be acting as a addictive drug as well, like opioid or cannabinoid or other addictive drugs, right? To answer that question, we have used another set of animals which are healthy in nature, the naive animals without any neuropathy. So if the animals which are healthy, which are not suffering from any pain disorder, if they also show the preference to burgeoning or any drug, that means this drug is having the addictive potential. So to our surprise, interestingly, the naive animals, the healthy animals, did not showed any preference to the burgeoning paired chamber, which clearly indicate that this increase in place preference for this chamber is associated specifically with the pain state of the animals when the animals are suffering from chemotherapy induced neuropathic pain. So it's pain state dependent. And then we calculated the difference score, which is another way of presenting this data, which clearly indicate that in CINP animals, the time spent in drug paired chamber was more as compared to saline paired. While in case of naive animals, healthy animals, there's no difference in time spent or difference score, you can say. This is the heat map of the animals, which is automatically given by the software. You can see during the preconditioning, the density of heat map in both, vehicle paired and burgeoning paired is almost same. But in the these are the naive animals, healthy animals, and in the post conditioning also, the heat map clearly indicate that the time spent in the vehicle and the burgeoning paired chamber is almost equal. But in case of animals which were treated with which are suffering from chemotherapy pain, their preconditioning heat map is almost similar between vehicle and the burgeoning paired chamber. While in case of post conditioning, these neuropathic animals are spending more time in the burgeoning paired chamber and they're spending minimal time in the vehicle paired chamber. This clearly indicates that the drug can treat the spontaneous ongoing pain. Now, there are other tests as well for the measurement of spontaneous ongoing pain in animals. If you look at the publications by Jeff Morgill, who is a big shot in the field of pain research and is a professor in University of McGill, Canada. So he has published very good work in the field of spontaneous ongoing pain, where they are video recording the animals and checking their facial expression, checking their grimacing and their motor movement and all other functions. So there are other measures as well, but this is also one of the widely used measure for the measurement of spontaneous ongoing pain in animals. I think, how much time do I have? So,
Sir, it's up to you, sir. No
okay,
issues,
okay,
sir.
okay. Thank you. So I'm almost done otherwise. So this is the, okay. So in this data, if you look, we have repeated the same test with the another drug, which is gabapentin, standard drug, and we found a similar kind of data. The gabapentin was able to, show, more time spent in the gabapentin treated chambers. Then we repeated the same experiment, but this time with morphine. Morphine is one of the, I would say, most potential drug in relieving the pain. But unfortunately, morphine is suffering from a lot of side effects and toxicities, including drug addiction. That is the reason why morphine-like drug has been degraded from first line of therapy to second and third line of therapy. And we use other drugs like anticonvulsant, gabapentin, etc. these days. But so when we did the condition place preference by using morphine, we found that the animals, which were suffering from chemotherapy-induced neuropathy, there was a substantial increase in the time spent in the morphine pair chamber, and the time spent in the saline pair chamber was significantly decreased. And when we repeated this experiment in the healthy animals, in the naive animals, again we observed the same kind of behavior. So this means morphine does not distinguish between the neuropathic state and the healthy state, which indicate that morphine-like drugs are Definitely, they are a good analgesic. They can relieve the pain, but at the same time, they also produce the drug addiction because they are producing the strong place preference in case of healthy animals as well. Here you can see these are neuropathic animals and these are healthy animals. So that is the issue with morphine-like drugs. Here you can see there is a clear cut, increase in heat map density in the, both in the naive animals and in the neuropathic animals after the treatment with the morphine. So this is another kind of control in our study, which indicates that burgeonin does not produce this kind of behavior. It is selective only for the neuropathic animals and points that it does not have addictive potential, unlike morphine. Then we have measured the expression of different TRP channels like TRPV1, and then we found that burgeonin at a lower dose does not have any effect on the TRPV1 mRNA expression in the spinal cord of animals. But yes, at 1500 mg per kg, it significantly downregulates both the TRPV1 mRNA expression in DRG and spinal cord. Then the similar kind of effects we observed in case of TRPA1 mRNA expression, then TRPM8 also. But in case of TRPM8, if you look at this data, interestingly, we did not found any significant decrease in the TRPM8 mRNA expression, both in the DRG and the spinal cord, while gabapentin was able to, downregulate the TRPM8 mRNA expression. But interestingly, burgeonin was not having any effect on, on TRPM8 mRNA expression. So I did not put that in silico computational data here, but we do have the data which clearly suggests that burgeonin had very good interaction with the TRPV1 and TRPA1 receptors, but it does not interact very well with the TRPM8, which might be the reason why it is not able to downregulate the TRPM8 mRNA expressions. Then we have measured the NR2B mRNA expressions, which is a subunit of the glutamate receptor, and TRPA1 protein expression as well, and found that both of them were significantly elevated in the neuropathic animals, in the DRG and in the spinal cord as well. And when these animals were treated with burgeonin at different doses, 25, 50, and 100, there was a significant downregulation of the NR2B and also the TRPA1 protein expressions. Then we also measured the expressions of TRPV1 and the marker for microglia activation, and then ICAM1, which is a marker for blood-brain barrier integrity. We found that all these expressions were significantly upregulated both in the DRG and spinal cord of the animals which were suffering from chemotherapy-induced peripheral neuropathy. And when these animals were treated with burgeonin at different doses, we found that at low dose, burgeonin does not had any effect, but at medium and the higher dose, 1500 mg per kg, burgeonin was able to downregulate the expression of TRPV1 and IBA1 and ICAM1. So that's all. This is the overall summary of this work, where we found that when the animals were treated with combination chemotherapy, which is a combination of 3 anti-cancer drugs, paclitaxel, cisplatin, and vincristine, there was a increase in free radical generation due to TRP channels activation in the peripheral terminals, which lead to the increased influx of the sodium and the calcium inside the cell membrane, which further leads to the activation of NR2B subunit of the NMDA receptor, which further leads to the upregulation of pro-inflammatory cytokines, increase in macroglia activation, which further lead to the blood-brain barrier integrity compromise and lead to the infiltration of the pro-inflammatory cytokine from peripheral nervous system to the central nervous system, causing mitochondrial dysfunction, hyperactivation of the glutamate, release of NMDA subunit of the glutamate receptor, further leading to the macrophage T cell infiltration, and finally leading to the central sensitization and neuropathic pain. And when these animals were treated with burgeonin, it has prevented and blocked this signaling cascade and helps in relieving the neuropathic pain in these animals. So in discussion, I would just like to say that burgeonin was able to inhibit both evoked and spontaneous ongoing pain in animals which were treated with combination chemotherapy, and most importantly, burgeonin did not produce CPP in the naive rats, unlike morphine. which suggests that burgeonin does not produce addiction in these animals. Moreover, burgeonin also decreases the chemotherapy-induced upregulated mRNA and protein expression of NR2B, TRPA1, and TRPA1. And most importantly, it does not lead to any CNS associated side effect. That data I did not put here, where we have performed the rotarod studies to measure the motor coordination of the animals. And then we also performed the open field test where we are measuring how animals are moving within an open field arena after giving the burgeonin and morphine. So we found that after treatment with burgeonin, animals did not produce any sedative like behavior. And overall, we found that burgeonin could be used as a potential therapeutic for the treatment of chemotherapy-induced neuropathic pain by using this new animal model. This was the paper which was recently published in International Immunopharmacology. And these are the key findings, where, we are very proud to say that we have successfully established a new animal model of chemotherapy-induced peripheral neuropathy, which is clinically relevant as compared to the existing animal model, and also more robust and reproducible in nature. And we also, dissected the crosstalk, which is involved in chemotherapy-induced peripheral neuropathy, encompassing TRPA1, TRPA1, and NR2B crosstalk. And overall, we suggest that this study may provide a critical insight that will lay down the basis for future studies investigating role of TRP channels-based therapeutics for the treatment of chronic pain. So due to lack of time, I could not present another set of data where we have targeted TRPA1 selectively, because in this study, if you see that we have used one molecule, which is targeting different TRP channels, but there are always, challenges in terms of non-selective targeting of any mediator. So in another set of study, we have prepared a siRNA nanoformulation for targeting TRPA1 selectively. But that set of data I could not present today due to lack of time. Maybe some other day we can, talk to you again. So with that, I would like to, end up my presentation. And I would like to convey my thanks to Dr. Rajo, who is my postdoc supervisor, for continuous discussions and brainstorming sessions and helping us, and most importantly to the students who are the real contributor to the work which I am presenting today. So here you can see with me Akhilesh in the red shirt. So this all work belongs to Akhilesh. He has conducted his work, all this work during his PhD, and he has recently completed his pre-PhD seminar as well. So this is the PhD research work of Akhilesh which I am presenting here today in front of all of you. And then here you can see Somesh is here, another PhD student in the lab. Then Obal is there, Deepak, PMRF student in our lab, PhD student all, and Anaga is there, Meghna is there, and Priyanka, Deepa, all other master students we have, So new people have been added in the team. I don't have the recent photo, but thanks to all the students for their hard work. And I'm also thankful to IIT BHU Varanasi for providing the infrastructure so that we could have carried out all this work. And many thanks to all the funding agency, including MHRD, Indian Council of Medical Research, Science and Engineering Research Board, PMRF, and UPCST for supporting our lab with funding from time to time. And thanks to all of you for your patience, for bearing with me for almost more than 1.5 hours. And if you have any questions, feel free to contact organizers, write to them, and once I hear from them, I will try to address your questions as much as I can. Thank you so much. But before you leave from here, always remember, pain is temporary. It may last a minute or an hour or a day or a year, but eventually if you don't take care of your pain, something else will take its place. So please be aware of your pain and take care of it. Okay, thank you so much for your patience. And once again, thank you so much to the organizing team, Dr. Alok and Dr. Monica and Dr. Sanjay for inviting me for this wonderful session. Thank you.
Thank you so much, sir. There are two general questions which I would like to ask you. The first is, what ethical considerations guide the use of animal models in neuropathic pain studies?
You mean to say what ethical considerations?
Yes, ethical considerations. Yes, sir.
So, whenever we talk about the ethics, there are always a lot of concerns, like if we talk about the animal ethics you are talking about, I think, right?
Yes, sir. Animal ethical
Yeah.
committee.
So definitely, whenever we do any kind of animal studies, animal ethics is always the primary concern. So doesn't matter which animal model you are going for, you always have to make sure that ethics should never get compromised. But yeah, in this model specifically, we are using the toxic drugs which are anti-cancer in nature and having a lot of side effects and toxicity as well, right? So we had made sure that we are providing the best care to these animals in terms of feed, water, their bedding, time to time care, measuring their vital parameters, and making sure that they do not suffer during the entire study. So ethical concerns are always very, very important whenever you do any kind of animal studies.
Okay. So one more question is there. Are there any particular challenges in translating findings from animal models to human patients?
Yeah, of course, that's a very good question actually. So as I mentioned my first few slides as well, more than 90% or 95% of the drugs, which you know completed the preclinical phase successfully, they do fail in the clinical trials. And there are a lot of reasons for that. One of the reason I explained, is because of the choice of the wrong animal model. But again, we are working on entirely different species. When we talk about the comparison between in vitro model versus in vivo model, then also we see a lot of differences, because in the in vitro model we are working on specific cells, isolated population of the cells. But animal system is a complicated system, it's a complex system, right? It's a combination of different organ system as well. So a drug molecule behaves entirely different in vitro condition versus in vivo condition. But then when we changes the species from rodents to humans, or large species like monkeys, dogs to humans, these are again two completely different system. So there are genetic differences as well. There are other pharmacokinetic parameters, or differences, which accounts for I would say drug failures. So that's why recently the field of organoid is emerging very, very quickly, where we are developing a organoid system, which is a little bit complex system, which is I would say higher version of in vitro system but lower than the animal version. So we can make the human organoid where we can use like combination of different organ system and see how a particular drug or molecule is behaving in that system. So that is to the humans, I would say. But
Yeah.
yeah, there are always challenges when we talk about the
Okay. Sessions. Okay.
testing of new molecules.
Thank you so much, sir. Then let's moving towards the vote of thanks for the concluding session. In conclusion, our journey today has been a captivating exploration from the laboratory to life, delving into the intricate realm of unraveling chemotherapy-induced neuropathic pain with cutting-edge animal models. The insights shared by our distinguished speaker, Dr. Vinod Tiwari, sir, have illuminated the path forward for researchers, practitioners, and stakeholders alike. As we navigate the complexities of neuropathic pain, the bridges built between the laboratory and real life applications become clearer. The knowledge exchange today, coupled with the collective expertise of our global audience, holds the promise of advancing both our understanding and potential interventions in this critical area. And the commitment of transformative research exhibited by Dr. Tiwari sir and echoed by our vibrant community serve as a testament to the power of collaboration and innovation in the field of pharmaceutical sciences. The wealth of perspective shared today, spanning more than 15 countries, underlines the global importance of addressing the challenges posed by chemotherapy induced neuropathic pain. We extend our sincere gratitude to Dr. Shashi Alok sir, National President SPSR, for gracing us with his esteemed presence today. In acknowledging your dedication and leadership, we express our heartfelt thanks for your invaluable contributions. Your presence adds immense value to our collective pursuit of knowledge and excellence in the field. In extending our deepest gratitude, Dr. Vinod Tiwari, sir, and our convener, Mrs. Monica Sabarwal, ma'am, and all our participants, we recognize the collective effort to push the boundaries of the knowledge. Let this webinar serve as catalyst for continued dialogue, collaboration, and action as we strive to translate cutting edge research into tangible improvements in the lives of those affected by the neuropathic pain. Thank you for being an integral part of 32nd SPSR webinar, and we look forward to future endeavors that contribute to the betterment of healthcare and well-being for all. Have a wonderful day. Thank you. Thank you so much, sir.
Thank you so much, Dr. Sanjay, one more time. And I would request you to kindly share if there are any queries by the participants from different parts of
Sure,
the
sure,
world.
definitely, sir.
And also, if there is possibility, kindly share the feedback as well, feedback of the participants.
Sure, sure, sir. Sure.
Okay. Thank
Thank
you.
you, sir.
Thank you once again.